So here we have 3 cells in series, okay? And the information will go from photoreceptors to the bipolar cells to the ganglion cells, and it's the ganglion cells axon that becomes the optic nerve. What type of neurons are going to be found in our retina in terms of structure? So it's going to be bipolar neurons, right? It's going to be bipolar neurons. So these are, even the photoreceptors are bipolar neurons, right? These 3 are all bipolar neurons. So we'll look at them as well. Okay. But the information, it's not going to say action potential. It actually is not action potential produced here. Information goes from photoreceptor to bipolar to ganglion cell. Okay. Now I'll show a image of orientation. For our course, we want to really know how the transmission of the impulse is. Okay, so 3 cells. Information is going to be transmitted from the photoreceptors to the bipolar cells to the ganglion cells. It's the ganglion cells that produce the action potential. It's then that becomes the optic nerve. What is the optic nerve? Found posterior, yeah. What else can we talk about? Is it a cranial nerve? What number?
2.
2. So it's going to take what type of information? Visual sense information. Right? So it's going to take visual sense information from retina back out. We'll talk a little bit more about this. So in our retina, we have 250 million photoreceptors. Right? So lots of them. This is again, we mentioned about 70% of our total body's receptors, sensory receptors, are in the eye. Right? Here is where it's mostly concentrated. We do have 2 types of receptors. We need to recognize the first name and what their functional or how they're different. Okay. Before that, we'll talk a little bit about more of the structure of retina. So fovea centralis. So if light passes through, the middle of the light will hit this spot. Okay. Fovea centralis contains mostly. Does anyone know which one's better for sharp vision? Which one's the one for color vision?
Cones.
Cones are color vision, rods are not. It's more grayscale. Sharper visions are also the cones. Okay? So they are concentrated in the center. So we're concentrating on something, that image is what's falling, what you want fall from the fovea centralis. You may have also heard macular lutea or macular degeneration, right? This is in that area. So fovea centralis is right in the middle of macular macular lutea. Sorry, that's like if you think about as a kind of a dartboard, the bullseye would be the fovea centralis, just cones. We go further out, the number of cones is still high, but it's going to reduce, and after a certain place, there's really no cones. It's just rods. So where the optic nerve leaves the eye, that image show, no, I have a better image. Sorry, that one was the one I was whereas optic nerve leaves the eye, there are no photoreceptors. Okay, and we all have it. We have what's called a blind spot, and we will test it in lab. It's kind of cool and kind of freaky because you see something and then you don't. Okay? It's there. It's in our eye. Why when you're looking at me, when I'm looking at you, then I don't see holes in us? 2 reasons. Yeah, they're working together. So what if I close one eye? How come I still don't see? If you close one eye, you're gonna see it. Yeah.
Your brain fills in the spots?
It does. Your brain does. Okay, so we will make sure that it doesn't in lab. We'll test it out. Kind of cool and kind of freaky. So as we put it on the optic disc, which is the blind spot, and you will not see it. Okay. So if I ask what is the blind spot or what is the blind spot? It lacks photoreceptors, This is the optic disc, that's where the optic nerve is leaving. There are no photoreceptors on this spot, hence it's a blind spot. Yes, light does strike it, but nothing is registering, right? Retina, given that it's got a lot of photoreceptors, lots of these receptors, we need a lot of nutrition, there's a lot of turnaround. We get some blood from the choroid that we mentioned. What's the choroid here? The middle layer of the eye that provides nutrition for all three layers. So we get some nutrition from there. We also have a blood supply dedicated just to the retina. Comes with the optic nerve, the central artery, and then spreads to the eye. Okay. So it does have a good blood supply because we want a good blood supply to the retina. Here's the image that I was trying to talk to. So here again, similar view, sagittal section, but we're looking profile view. This is anterior, that's posterior. That will be the cornea here, lens, okay? So light has to pass through the cornea, through the aqueous humor in the anterior chamber, pass through the opening in the iris, hit lens, and then go through the vitreous humor in the posterior chamber, posterior cavity, and then hit the retina. This image is a little bit of a magnified here. So light is still coming from the left to right. We're looking at this image, okay? Let's talk about the optic nerve first. So here's the optic nerve leaving, the yellow fibers. This is to show the retina, the three cells we'll talk about in a second. It's not present here. So if any light hits this spot, they will not be recognized. They will not be sensitized. Okay. In terms of the layers here, we mentioned how the inner layer is the neural layer, right? Because light's going to hit them first. Okay. In this one, I don't think it's a great image. There's a little beige yellow color between these colored cells and the red layer here. You can just about make it there if you kind of picture it, it's there. Okay, that will be the pigmented layer of the retina, and then the red here is for the choroid, and then what's this white at the back here? Sclera, right? Covers the entire, well, not the entire of our eye, but it covers most of our eye without the cornea. So light's going to come through the front, so it really passes through the inner layer and it comes afterwards, right? So we have pigments in the pigmented layer of the retina that will absorb some of the light, and then to make sure nothing passes back, the choroid will absorb the rest of the remaining light rays. Here's a fundoscope. It's an image if they dilated the eye and took a picture. This is what they would see, or similar to this. That white area is the where the optic nerve leaves. That's the optic disc. There are no photoreceptors there. And here they've also shown, you can see a darkened area that's got a high concentration of cones. That's macula lutea. Right in the middle of that is the fovea centralis, which just contains purely cones. You can see the blood vessels, right? Thicker versus thinner. Which one do we think is the artery? Thicker one's a thicker wall. We'll learn about that in the cardiovascular system. So let's talk about the photoreceptors. Rods, those are mainly for grayscale, but they do not need a lot of light to be sensitized. Okay, so even in dim light, they can be sensitized. That's why it says very sensitive light. Even in small amount of light, they would be stimulated. Okay? So they're best suited for night vision and then peripheral vision because we do not have any cones in the periphery of our view. Okay? Single pigmented, they only give gray tones, and we'll talk about this in a second. We have a don't know if I include that mention. You can see multiple rods that will connect to one ganglion cell. So image falls on these multiple spots, the brain does not receive them as multiple spots, but as one spot. So that's not concentrated. Whereas cones, it's one cone to one ganglion cell. Okay, so its information is more precise. Convergence talk about how multiple rods will converge to a single ganglion. So in terms of the cones, unfortunately, yes, they're the most highest acuity, but you need bright light for them to be sensitized. Okay? So if you are not in your own home, but in a different place with very dim light, you would not see anything in color. Okay? You generally see in gray tones. Your brain puts it in when you're at your home, so you may think you know what the color of this, the furniture around your place is, so you probably will see it in that sense, right? But in dim light, okay, we only see gray tones of it. That's because the rods are insensitized. The cones do not work with any bright light. So they do have multiple pigments. We have 3 pigments in the cones. This is what helps us to distinguish different colors. If one of those pigments is not present or non-functional, that's when we develop color blindness. Okay? And in terms of their pathway, it's non-converging. So one cone transmits the information to one ganglion cell. High detailed, high resolution image we get. Macular lutea is more in the central region. It's more like a bullseye, periphery of it, if you will. And then right in the center is fovea centralis, just cones. That's the best spot to be. If you're concentrating on something, that's what it's going to form the sharpest image possible. And I mentioned color blindness has to do with either the absence or non-functional pigments on those pigments. So this is what if there was a doubt that somebody was colorblind, they'll use what's called the Ishihara charts. It was a Japanese physician who described it, right? Anybody think we're colorblind? Sure, you don't have to. In lab, if you wish to, it would be useful for your table mixing. I had a lab colleague who was colorblind, so it was interesting to compare what the Ishihara charts look like, right? Hopefully we all see the 16 here, right? What do we see here? 2. If you squint carefully or pay more attention, you might see the 8 there as well, okay? Those are individual. If we are colorblind with red, you would have seen that 8 much faster, okay? Kind of will see that connection. So the red on red helps. So it does make a difference because if they do not have that pigment or pigment that recognizes the color, they do not see that color as that color red. They just fill it out as another color. So this is a nice chart that gives a nice comparison. Rods, non-color, only one visual pigment, gray tones only. Here, color vision. We have 3 pigments, hence able to see. Rods are highly sensitive to light, so even the littlest light they will be triggered, while cones need more light. They are low sensitive. Low acuity, so that's the resolution, if you will. It's not good with rods because multiple rods converge to one ganglion cell, while higher acuity with our cones, with one cone, one ganglion cell. In terms of number of cells themselves, this one outnumbers the cones. Okay, a lot of periphery, only a small macular lutea. Okay, they're found mostly in the periphery, here mostly in the central visual field as well, because of recognizing the central location of those cells in the retina. So let's talk about refraction. So what's wrong with this straw? It's broken? No, right? That's the bending, the refraction of light. So here it's water and then glass. So here it's just passing through air and glass. Okay? Generally glass, I mean, it can vary, but here it doesn't look broken because it's only passing the air and glass. Here it's passing through the water and glass, so they bend it at different angles. Okay, so bending of light is called refraction. Okay. In our eye, please recognize two places or two structures which refracts it. What were the two places? You mentioned it earlier. Which one focuses the light? The lens does, as well as, what did I say, has the higher resolution, higher refraction. It's our cornea. Okay, so it's cornea and where is it? I didn't put it here. It's the cornea and the lens that have the refractive power. It's really the cornea. Both them do. The cornea has a higher refractive power compared to the lens. And the lens helps to focus the image. Okay, it's the convex nature of it. Focus the image. And it's similar. I think I mentioned this when we performed our microscopy. The images that are actually formed are not in the same orientation, right? We remember the letter E. How did it look?
Upside down.
Upside down and
backwards.
Left and right also. Okay, that's the same thing that's happening in our retina too. The image here will fall, so it's up on top, and to my right, it will fall on the bottom. And the flip upside down as well as right side. Pass through that direction. Okay. So how come we don't see each other as high up there? The light actually is coming. Yeah, brain. So it's upside down and reverse right to left. So in terms of the light passing through structures, right? So it's going to pass through the cornea. It's passing through the aqueous humor. Where is the aqueous humor found? It's the anterior cavity. Then it passes through the lens that divides the anterior cavity and the posterior cavity. Vitreous humor is found in the posterior cavity. And then it hits the retina, it hits the neural layer. I didn't emphasize, but if you look at the cells, what were the three cells in our retina? Neural layer. So it's photoreceptors, bipolar cells, ganglion cells. So here, the blue and yellow cells are the photoreceptors. The red is the bipolar cells, and then the greenish gray is the ganglion cell. Okay, so information, light is going to go pass through the entire layer, hit the outside of the retina, outside of the photoreceptors. Okay, and then information is going to go from them to the bipolar cells, to the ganglion cells, and then go as through the optic nerve. Okay, that's what this is talking about. Talks about the entire neural layer of the retina before it hits the photoreceptors. I mentioned the two structures that will refract the light, the cornea, the lens, bending of light is refraction. And the bigger refractive power is with the cornea, which is why laser treatment. Then lens helps with the focus. Any questions so far? Okay, so let's talk about, I think I mentioned this, why is best vision called 20/20 vision?
Pretty much it's what normal people see at 20 feet and what you see at 20 feet. It's the same.
Kind of, yeah. Elaborate.
Um,
It has to do with 20 feet.
It verifies and measures the, amount that your corneal lens starts focusing.
Mhm.
And it makes sure that the light that is put through to the retina, when you're focusing on something at 20 feet, isn't adjusted from what a normal person's would be.
Yeah,
If
good.
that makes sense.
Yeah. So it's basically our eye, in a most relaxed state, will be able to focus an object 20 feet away. So 20 feet will fall perfectly on our retina. And then again, we compare that. That's why it's not 100, right? We like 100%, but here they did not use 100 or 100. They used 20, but it talks about that reference. Where the most relaxed eye is really supposed to be suitable for distant vision. So that's 20 feet away. Okay? That's what the emetopic means. 20 feet away, this object should be perfectly focused on our retina. Right? That's the 20/20 vision talk. At this time, the ciliary muscles are relaxed, the lens is nicely stretched out. Okay, it's when we are concentrating on something closer, it becomes more rounded. Right? But its lens is stretched flat at 20 feet away. So what happens when we are looking at something close by? Three things happen, which you might have guessed with the lens. What's it going to do?
Changes its shape.
What shape will it become? More rounded because it's stretched out flat. I mentioned that 20 feet, when you're concentrating something close by, it becomes more rounded. Okay? And then that's the accommodation part. Okay? But you may have also noticed when somebody else is reading, you're going to see that the two eyeballs are not still remaining like this, but they actually converge. Okay? So you're concentrating on something more closer. Third thing happens with our pupils, right? Will they dilate or constrict? Constrict because you're concentrating on something small. Okay? So pupillary constriction. The two eyes will converge. So you're going to look closer, not parallel, but more tilted towards the middle, as well as the lens changes shape, becomes more round during when you're looking at something close by. Okay. So let's talk about some refractive problems, right? So if we are wearing glasses, especially at an earlier age, I know they say it's short-sightedness, right? Basically, they're not telling what the problem is. They're saying what we can see. Okay? Short-sightedness, people who have short-sightedness, what do they have difficulty with? Seeing things further away, right? That's called myopia. Okay? Short-sightedness and nearsightedness basically have difficulty with focusing images too far out. I think I have an image to show you. It's a concave lens because the focal point is actually it's further in front from the retina. Okay, so what we want the rays to be a little bit further out, so we want them entering the eyes further out. Is what the concave lens does. This is not very often. Hyperopia we don't see very often, but it can happen. Here we say the eyeball is too short. Now the focal point is actually behind the retina. So we want the rays now to come in even closer. So it was going like that. We want them becoming closer so they will focus on the retina. Here we would use convex lens. But what happens as we age? You may have experienced it. You may see your parents, your grandparents.
You
What do
need
they
reading
do? So
glasses.
they're reading. They bring it here. No, they break it away, right? Because now it's difficulty in. It's similar to this, but this is not the reason. Okay, it's not the eyeballs too short. It's the muscles are becoming more lax. We cannot make it more rounded. So hence we are having to move it further away from the eye itself. That is different. I think I have But before that, astigmatism is, some people may suffer with it, that's the middle column. If you have glasses, you will see the prescription, there are 3 columns. The first one talks about generally myopia, it's a negative sign. I talk about the power of that. The middle one has to do with something to correct the astigmatism. You can think of astigmatism as not a smooth either cornea or the lens. It's a little bumpy. So when you think about rays hitting this bumpiness, will it go nice and straight? No, they'll get distorted. Yeah, so hence they need to be corrected through. So here are some images. So normally something at a distance should focus nicely on the retina. Someone who suffers with myopia, the focal point is too early. It's said that the eyeball is too long. Okay, so we want the rays to come in further out, hence they'll hit the same spot, right? So further out, we want a concave lens, which will feel thicker on the outside compared to the middle. Okay, so the rays will come from a further out position, so they get focused closer to on the retina. Whereas in a patient who suffers with hyperopia, their focal point is too far because it's said that the eyeball is too short. Here we want the rays to come even closer, so this spot will be here. Okay, so they will wear convex lenses. Any questions? So in terms of what happens, you might have experienced this too, when you move from an area of darkness to more brighter light, right? We kind of squint because a lot of light, pupils will what in the darkened area? Dilated. Now when you come to the bright light area, lots of light are going in, right? So our reflex is trying to limit that light coming through. Why we get a glare is because that light came suddenly, it actually hits our retina and breaks down some of the pigments. You may have noticed, especially at night, you're driving, somebody has their eye beams on, and then it hits you in the eye, you are blurry for a little while, right? This is because of those pigments. Because your pupils were dilated. So we can actually lose a good vision for about 10, 15 minutes even. Usually it comes back a little gradually because again pigments do get replaced all the time. So they're already under something, were under the process. So sometimes it takes about a minute. People get a good visual feel, but it's still not perfect, right? Because it has to adapt to that change, the loss of those pigments. Next, we're going to talk about the visual pathways. So, we're going to start at the retina. We're going to have the optic nerve. And I mentioned this, I forget, I think it was the cranial nerve chapter when we were talking about it. It might have been the central nervous system. It's going to do a little X, okay? But, what's one of the functions of our medulla oblongata? Yep, it controls our respiratory rate, it's our respiratory center, cardiac centers. What else? Lots of important. Vomiting centers, hiccuping centers, all those reflective centers also there. Some cranial nerves are there. And then another important one. Why does our right side of our brain control the left side of our body mostly? Because the fibers, 90% of them cross over in the medulla oblongata, right? We have a similar, again, I want to be careful saying it's the same, it's not the same. It's not all the fibers are going to get crossed. Only some of them do, okay? So only the medial fibers get crossed over. So, we're going from the ganglion cell, we have the optic nerve. We'll go to a structure called the optic chiasma, okay? At this spot, the medial fibers, the medial neurons that came from the medial aspect of our retina will cross to the other side. Maybe I should draw. visualize because that drawing shows it in different color. I took an image out of it. I'm going to use 4 different colors, just use 2, that's why I'm drawing to try and help me. Okay, so let's draw the eyeball. We draw just 2 colors showing the 2 sides coming from the retina. Just 2 colors. These 2 are the optic nerves that are coming to meet at the optic chiasm. Here, only the medial fibers will cross over, while the lateral fibers remain on the same side they came from. A couple of things we're going to know is, now this is not called the optic nerve, it's called the optic tract, okay? It's got fibers from this eye and that eye, okay? What's sitting on top, let me rephrase that. So this structure is found over sella tussica, okay? If this structure is found over sella tussica, a cancer, a growth of which organ could cause damage of this optic chiasm.
Pituitary gland?
Pituitary gland. If someone comes with a pituitary adenoma, that's swelling of the pituitary gland, it could squeeze this. What side of their visual field would be lost? Is it the middle or the lateral side?
Lateral?
It's the lateral side. Why? Remember what the lens does? It puts the lateral images onto the middle. It's going to make it left and right. And these middle fibers are bringing lateral vision. They actually will lose this vision. They'll look here and you show them any movements here, they cannot see it. It's virtually like they're blocked off from here to here. Okay? It's called bitemporal hemianal. They cannot see on the 2 halves of the temporal half because the pituitary gland squeezing this area and squishes the middle fibers. If you ever see a patient, you can bet your bottom dollar and put that as your diagnosis. They will scan it and they'll find the pituitary adenoma. I would say 99% of the time it's correct. There could be a bleed that could cause it, but most likely cause it. Okay, anyway, so this is optic nerve, optic chiasma, and the optic chiasma medial fibers crossover. Then we have the optic tract, okay? That will go to the thalamus, okay? Everything pretty much goes to the thalamus. From the thalamus, it's going to go to our primary visual cortex. Where is our primary visual cortex? The occipital lobe, right? So it goes to the occipital lobe. It's called optic radiation here on the side. Okay? But we do send information to other parts of our brains. We'll send to the midbrain. That's our reticular formation for our awakenedness, right? We'll send it for reflexes to superior colliculi, for example, we mentioned in the midbrain again, and different parts of our brain. See? Yeah, so we will send information to the midbrain. will also send to the hypothalamus sleep wake cycles, right? That will help to regulate that as well. Like during the day, which is why nowadays it might be difficult to wake up compared to in summertime because of the light dimmer outside. So here's an image showing the two sides, only the medial side crosses over. Okay, all the lateral side stays. It's optic nerve, optic chiasma, optic tract, and then optic radiation goes from the thalamus to the occipital. So we'll test this out. For depth perception, we need both our eyes, okay? Because both our eyes are focusing on the same object at slightly different angles. Okay, and our brain that puts the two together to get the three-dimensional image of an object. Okay? So you need for both eyes. We lose sight in one eye, but we generally will lose our three-dimensional vision. We may remember it from the past, or we could rotate our head